Dr. Sachs joined the FDA in 1998 as a medical reviewer in the Office of New Drugs, OND. His subsequent positions included acting director of the Office of Critical Path Programs and his current position of associate director for clinical methodology in CDER's Office of Medical Policy. In this current capacity, he has led efforts to support the use of electronic technology in clinical drug development. Besides his involvement in the design and analysis of clinical trials, he maintains a special interest in tuberculosis and other tropical diseases and has published and presented extensively on these topics. Please help me welcome Dr. Sachs.
Well, thank you very much, Brenda, for all the kind words. To all members of the audience, good morning or good afternoon or good evening, depending where you are. I'm Leonard Sachs from the Office of Medical Policy in the Center for Drug Evaluation Research at FDA. And I'm delighted to welcome you to this Investigator training course update. At FDA, we've long recognized that clinical investigators and others involved in the trial enterprise are amongst our most important partners in the development of medical products. We rely on you for all the clinical data we receive to support the approval of new drugs, biologics, and devices, and we are tremendously appreciative of the critical contribution you make to medical science and to the health of our society. Yet the opportunities to share our thoughts and experiences with you are limited. With industry and with trial sponsors, we have many opportunities for discussion. There are a range of meetings with FDA that sponsors can request at various stages of product development, but there is really no formal framework to meet with investigators. So recognizing this, we've considered it very important to reach out to you and to share our experiences in this interactive forum. As many of you know, we've been running this training course for well over a decade. In normal times, this has been a big 3-day event where we've met in person and covered a broad spectrum of topics related to the development of medical products. Typically, we've included lectures on non-clinical drug development, animal studies, toxicology, CMC, on clinical trials, statistical analysis, drug safety, ethics, and regulation. Last year, we were forced to cancel the course completely. But this year, we've decided to do an online update tailored to a virtual environment. So we've limited this to approximately 2 4-hour sessions today and tomorrow. And the idea is to cover the most topical areas of interest to investigators. As we know, the pandemic has presented challenges to all of us, and together with others, FDA has been very involved in trying to keep existing trials going. We published guidances on decentralized strategies and remote data gathering to save patients from having to come to clinical trial sites. FDA has also been immersed in the development of new products to prevent and to treat COVID, and several of these have been discovered, approved, and used in record time, saving countless lives. Today, we will focus on selected topics of new scientific importance. I'm sure you will all be interested in hearing about drug development programs for COVID, about novel drug development in cancer, which is currently one of the most active fields, and new technologies like CAR-T and gene therapy, etc. Tomorrow, we will focus on some of the innovative operational issues in clinical trials, including real-world evidence, drug repurposing, digital health technologies, and the regulatory requirements for investigators. So by sharing our experiences inside FDA, we're hoping to provide you with an insight into FDA requirements that will give you as investigators greater confidence and expertise in conducting trials, and that it will improve the clinical trial enterprise in general. We are thrilled to see the enormous interest in the update. As Brenda had mentioned, we have about 4,500 attendees enrolled today. I also want to thank our speakers in advance. All the speakers are colleagues of mine at FDA who have generously taken time to participate and to prepare these presentations. I will apologize in advance for not being the most technologically gifted, but I hope we will be able to give you a satisfying experience as free of glitches as possible. During the panel sessions, we hope you will send in your questions to make this as interactive as we can. So without further ado, I'd just like to review the agenda. We will begin this afternoon with a lecture on gene therapy by Lei Xu, followed by a lecture on CAR-T therapy by Lianne Hu. We'll have a panel discussion. This will be followed by Sandra Kasak presenting on innovations in the design of clinical trials in oncology. After that, we'll have a talk from Kirk Chantak and Sarita Boyd on COVID-19 treatment. There'll be a panel discussion after that, and we will end off with a discussion on trial populations, diversity, sex differences in pediatrics, with Matilda Fan-King, with Kavita Vasisht, and with Lin Yao. So without further ado, I'd like to turn over the microphone to Lei Xu. Thank you.
Thank you for the introduction. My talk will focus on the clinical development of gene therapy products. There are two learning objectives. First, grasp important principles on efficient clinical development of investigational gene therapy products. Second, understand the regulatory requirements and flexibility for marketing approval of those gene therapy products. We'll start with an overview of gene therapy, followed by efficient clinical development of gene therapy products. Then I'm going to talk about the regulatory requirements and flexibility. And finally, I will give an example of gene therapy development program. Human gene therapy products mediate their effects by transcription or translation of transferred genetic material, or specifically altering host or human genetic sequences. Here is the list of the common gene therapy products. There are two categories of gene therapy, ex vivo and in vivo. For ex vivo, the most well-known is this chimeric antigen receptor T cells or CAR T cells. In this type of products, the cells are taken from the human. The vectors are injected into those cells to make those cells genetically modified cells. Then these genetically modified cells are introduced back to patients to treat their diseases. For the in vivo gene therapy, the transgene carried by a viral vector or plasmid or bacteria are directly injected into the human body, and the transgene are expressed within the human body. And this chart showed the number of the new INDs for gene therapy products that are submitted to us over the last 30 years. And you can see over more than 2x increase over the last 5-6 years in regard to the numbers of new research INDs we receive each year. In 2017, we approved the first AAV-based gene therapy product, Voratigene, which is indicated for a treatment of retinal dystrophy due to mutations in the RPE65 gene. In 2019, we approved the second product on the Onasemnogene abiparvovec for the treatment of spinal muscular atrophy. And also in 2017, we approved the first genetically modified autologous T-cell or immune therapy products, Kymriah, and for the treatment of acute lymphoblastic leukemia in people younger than 25 years of age and large B-cell lymphoma. And subsequently, we approved 3 other products for similar indications. And in 2021, we approved another genetically modified T-cell immune therapy product, and it's for the indication of multiple myeloma in adults. Next, I'm going to talk about the clinical development of gene therapy products. And I included here the link to our cell and gene therapy guidances, and I also listed some of the important guidance documents for CMC, preclinical, and clinical. There are a lot of similar fundamental considerations between gene therapy products and other biological products regarding their clinical development program. The clinical development program can vary significantly for different diseases. And so we encourage sponsors to discuss with us early in the product development. For early phase studies, the primary objective is safety. Meantime, we encourage sponsors to also explore activity and preliminary clinical efficacy. And for gene therapy products for rare diseases, we encourage sponsors to hit a home run by designing the first-in-human studies to provide evidence of effectiveness, and also to resolve as many manufacturing issues as possible before the initiation of the first-in-human study. Next, I'm going to talk about study design. Randomized concurrent controlled clinical studies may be the most efficient means of obtaining persuasive evidence of effectiveness. Because many diseases, especially the rare diseases, have poorly understood etiology and or pathophysiology, and are poorly characterized or have highly variable natural history. So we encourage sponsors to consider randomized concurrent controlled double-blinded studies even for the first-in-human studies. Such design can facilitate data interpretation, and this is especially important for rare diseases because such design not only maximizes the use of valuable patient resources, but also it may provide sufficient evidence of effectiveness to support a future marketing application. For such a design, we encourage sponsors to allow all the subjects to receive standard of care. No subjects should be denied effective therapies to be randomized to a control arm. And we encourage sponsors for an add-on design, which means all subjects receive the standard of care, then be randomized to either the gene therapy arm or the control arm. Next, I'm going to talk briefly about external controls. External controls may be appropriate if all of those following criteria are met. There is an unmet medical need for the condition to be investigated. The concurrent control is not practical or ethical. The disease course is well documented and highly predictable. The study population and historical controls are suitably comparable, and the effect from the treatment is expected to be large, self-evident, and temporally to the intervention. In general, it is challenging to meet all the above criteria, so historical control may not be adequate. So generally, use of external historical control in place of a concurrent control is not encouraged. Next, I'm going to talk about dose selection and product delivery. Early phase studies, we recommend a dose range study, and the initial dose should be supported by preclinical studies and all available clinical information. The dose should be reasonably safe and also have a therapeutic potential. And we encourage substantial dose exploration throughout the clinical development to identify a safe and effective dose. And this is particularly important for certain gene therapy products which currently do not allow a second administration because of the immune response that was generated following the first administration. A lot of gene therapy products are delivered using invasive surgical procedures. And to protect the patient's safety, we recommend a staged approach, which means inject the product to one side first for the first several patients. If the safety profile is reasonable, then the product can be administered bilaterally. And also, we require that a detailed description of the delivery procedures and the list of the devices used be included in the clinical protocol. Monitor the risks associated with the gene therapy products, and the administration procedure include standard safety monitoring, and also monitor for immune responses which include cellular and humoral immune responses to both the vector and the transgene coded protein. And also for some products, monitoring of the potential insertional mutagenesis is necessary. The duration of the monitoring should be sufficient to cover the expected duration of the effect. And it should be dependent on the information from preclinical studies and experience with related products. Long-term follow-up is required for some gene therapy products. For example, 15 years of follow-up for integrating viral vector-based products, such as those CAR-T products. And clinical development can continue while long-term follow-up of those early phase study project subjects is ongoing. Regarding the study endpoints, for early phase studies, the primary endpoint is safety. Meantime, we encourage the sponsors to explore a broad range of endpoints, include clinical endpoint to assess a potential clinical benefit, and the biomarkers to assess potential activity of the gene therapy products. And all those can help further guide the clinical development. Endpoints in the trials to support the future marketing application. The primary efficacy endpoint should be either clinically meaningful endpoints, which directly measure how patients feel, function, or survive, or surrogate endpoint, which is reasonably likely to predict clinical benefit. Next, I'm going to talk about regulatory requirements and flexibility. Approval of the drugs and biologics must be based on substantial evidence of effectiveness and evidence of safety. And the evidence of effectiveness should be obtained from adequate and well-controlled studies. We understand that certain aspects of product development that are feasible for the common conditions may not be feasible for rare conditions. So our regulations do provide flexibility in applying the regulatory standards for those rare conditions. For the evidence of the effectiveness for rare disease, there is no specific minimum number of the patients to be studied to establish the safety and effectiveness of a treatment for any rare conditions. The number of the patients needed to establish the effectiveness and safety is determined on a case-by-case basis and taking the following into consideration: the persuasiveness of the data, the nature of the benefit, the patient's population that would be treated after approval, and the concerns for potential of harm from the treatment. Next, I'm going to talk about an example of the gene therapy program, Luxturna, or variety gene depovovac. It is the first-in-class AAV vector-based gene therapy that was approved in 2017. It is delivered using the subretinal injection procedure, and it is indicated for the treatment of retinal dystrophy due to RPE mutation. It is a rare condition with about 1,000 to 2,000 patients in the U.S. The clinical manifestations may vary. It usually starts with night blindness and progressive visual field loss. Complete blindness will occur in all patients later in their life, and there is a significant impairment in their activity of daily living. There was no approved pharmacological treatment prior to the approval of Luxturna. So the picture on the left shows a normal vision, and the picture on the right shows the patients they have a decreased light sensitivity. So what they see is very blurry and dark. So we worked with the sponsor to design and validate a new efficacy endpoint called multiluminance mobility test, which assess the patient's ability to mobilize at different light levels. And this endpoint was used in their phase 3 studies to support the marketing application. So this slide just shows one of the 12 courses of this test. So patient start from the start, walk along the arrow, and try not to hit into any of those obstacles, and all through the end. And the different light levels are used. The lowest light level the patient can navigate without any mistake was considered the success. And as I mentioned earlier, the phase 3 study used this as its primary endpoint, and there is a statistically significant difference between the treatment arm and the control arm. So in summary, during the development of the Luxturno program, several tools were used to facilitate its development and approval. This includes the breakthrough therapy designation, the priority review designation, and also the use of the novel endpoint and the non-traditional clinical development program, because they did a phase 1 study and then they did one phase 3 study, and both studies were used to support its safety, but the evidence of effectiveness was based on the single phase 3 study. So in summary, we believe it is critical to collaborate among different stakeholders to expedite the development of a gene therapy product. We encourage the sponsor to plan ahead and to design the early phase studies for rare diseases in a way it can provide the evidence of effectiveness and safety. So we encourage early, concurrently controlled, randomized, blinded study. We also encourage early communication with us. So now come to the first challenge question. So a concurrently controlled randomized first-in-human study is not necessary or recommended because the objective of such a study is to assess safety. True or false? Most of you get this right. It is false. We encourage such concurrently controlled randomized first-in-human study, especially for the rare diseases. So the second challenge question, which of the following can be used as a gene therapy vector? A, plasmids, B, lentivirus, C, adeno-associated virus vector, D, all of the above. And you are correct. It's D, all of the above. So this is my contact information. I also included my office and CBER's information. And thank you for your attention.
Good morning, good afternoon, or good evening to everyone. Thank you for attending the Clinical Investigator Training course. My talk today will be focused on clinical considerations for the development of chimeric antigen receptor T-cell therapies for cancers. My name is Lianne Hu. I am a clinical reviewer at the oncology branch at the Division of Clinical Evaluation and Pharmacology Toxicology under the Office of Tissues and Advanced Therapies at the CDER. This talk includes opinions from me and my colleagues at the oncology branch, but the opinions do not represent FDA policies, and I will not discuss off-label use. This is my disclosure. I have no conflict of interest. In this talk, I will cover 6 main topics. First, I will introduce how oncology products are regulated at the FDA. Then I will provide a brief overview of CAR products currently under IND investigations and in the global research pipeline. After that, I will summarize toxicities associated with CAR T therapies observed in clinical trials and some strategies to reduce toxicities and improve efficacy. However, most of this talk will be focused on number 5, clinical considerations during early phase CAR T product development. Lastly, I will introduce meetings that sponsors can request to communicate with the FDA. Now, let's look at the regulation of oncology products at the FDA. Oncology products are regulated by 3 different centers at the FDA. CDER regulates drugs which are small molecules. CDER also regulates monoclonal antibodies, therapeutic proteins, cytokines, and antibody-drug conjugates. Most relevant to CAR T therapies, the Office of In Vitro Diagnostics and Radiological Health at the CDRH regulates in vitro companion diagnostics, which are often used for study eligibility determination. CBER, where I work, regulates cell and gene therapies, oncolytical viruses, therapeutic vaccines, and microbiotas. On the very left of this slide, you see the name OCE, which represents the Oncology Center of Excellence. OCE oversees the regulation of all oncology products. Review at the FDA involves a multidisciplinary team. The clinical pharmacology, toxicology, and product teams are 3 teams that frequently work closely together in reviewing early phase trials. The clinical pharmacology team may also be involved in the review of clinical pharmacology components. The statistical team is an important review team, particularly in late phase trials. The regulatory project management team is our central channel to communicate with sponsors and organize meetings. As of now, we have received over 230 research INDs for CAR products. Most of them are CAR T products, but there are also a few other CAR products, such as CAR NK and CAR macrophages. Almost all of those INDs are cancer therapies. Early this year, about 86% of CAR products are autologous products, and 14% of them are allogeneic products. The proportion of these allogeneic products has slightly increased since January. Next, we will have a brief overview of different types of products and major components of a CAR construct. You may have seen this figure in the cited publication. It's truly amazing how fast the field of adoptive cell therapies is growing. As of April of this year, there were 2,073 active cell therapy agents in the global cell therapy pipeline. CAR T-cell therapies continue to dominate the pipeline with 1,164 studies, which is 299 more than the year of 2000. The figure on the left of this slide shows the most common targets in the hematological malignancies. CD19 and B-cell maturation antigen BCMA continue to be the top targets. The figure on the right shows top targets in solid tumors. We have seen many of those top targets in the INDs routed to our oncology branch. I will use the next 2 slides to say a few words about 2 types of CAR T products, autologous and allogeneic CAR T products. One can imagine that the logistics of manufacturing CAR products is very different from conventional therapies, since the manufacturing process starts with PBMC from individual study subjects or healthy donors Each step of the manufacturing should be designed for understanding how it will affect the final product attributes. Since the vector is critical to the CAR T cell function, full CMC information to support the transgene vector is expected. For autologous products, the chain of identity as well as treatment schedule is integral to the manufacturing process. Similar considerations are important for allogeneic products, but in this case, the focus is on donor eligibility and additional components, such as genome editing components to reduce alloreactivity, such as GVHD. If any of you is interested in regulatory considerations of manufacturing issues related to CAR T products, you can review a presentation by Dr. Kimberly Schwartz from CBER OTAC available on YouTube. I can add the link to the chat box later after the talk. This is a figure from a review paper published last year in Nature Review. It nicely illustrates the major components of a CAR construct and additional modifications researchers have tried to enhance the design. Starting from the left, CAR has 4 major components: an antigen binding domain, a hinge, a transmembrane domain, and an intracellular signaling domain. The antigen binding domain is usually a single-chain variable fragment and usually derived from a monoclonal antibody. The intracellular signaling domain generally contains a co-stimulated domain such as CD28 or 41BB, as well as T-cell activation domain derived from CD3 theta chain of the T-cell receptor. To further improve CAR design, an armor protein can be added to a CAR construct, typically on cell surfaces or secreted as immunomodulatory proteins, which enhances T-cell function or improve tumor microenvironment. In addition, gene editing of engineered T-cells is also possible to further enhance CAR functions. This slide summarizes FDA-approved indications of 5 CAR T therapies. The first CAR T therapy is Kymriah, approved in 2017. Abecamar, also called ide-cel, approved in March 2021, was the most recently approved CAR T therapy. The first 4 approved CAR T therapies target CD19 on B cells and are indicated for refractory B-cell lymphomas. Abecamar targets B-cell maturation antigen and is approved for refractory multiple myelomas. Now I will briefly summarize toxicities associated with CAR T therapies. Toxicities shown on this slide are described as box warning or in section 5 or 6 of the FDA labels of the approved CAR T therapies. They have also been reported in ongoing IND studies with CAR T therapies. Cytokine release syndrome is a well-established adverse reaction of CAR T therapies. It happens in 70 to 90% of patients. It is potentially life-threatening or even fatal. Neurotoxicity, particularly immune effector cell-associated neurotoxicity syndrome, also known as ICANS, is a neurological disorder following immune therapies. Infusion-related reactions can happen too. Patients should be pre-medicated with antihistamines and acetaminophen. Serious infections and prolonged cytopenia may occur and are generally related to lymphodepletion. Antimicrobial and antifungal prophylaxis should be given according to institution standard practice. Prolonged B-cell aplasia is a type of on-target and off-tumor toxicity observed in patients who received CD19 targeted CAR T therapies. Tumor lysis syndrome is not common, but it happens. We frequently ask the sponsor to address potential on-target off-tumor risk and have a safety monitoring plan including on-target off-tumor toxicities. Now, I will summarize some of the strategies to reduce CAR T therapy-related toxicities and improve efficacy. Here on this slide, the box on the left has listed the strategies to stop inappropriately activated CAR T cells and reduce CAR T cells related severe toxicities. One strategy is to add an inducible caspase-9 as a safety switch gene to induce CAR T-cell apoptosis when necessary. Another strategy is to add elimination genes such as CD20 to the CAR construct so that anti-CD20 monoclonal antibody rituximab can stop CAR T-cell function when necessary. The third strategy is to add cytokine antagonist or knockout cytokine genes to alleviate cytokine storm. The box on the right gives examples of strategies to improve CAR T efficacy. One strategy is to target multiple tumor-associated antigens to counter an antigen escape phenomenon. Second, to enhance the persistence of CAR T cells, one promising strategy is to select memory-like CAR T cells. Third strategy is to increase tumor homing and penetration. For instance, engineer CAR T cells to secrete tumor extracellular matrix modifying enzymes to target solid tumor structure and facilitate tumor penetration. This seems to be very important for the challenges we are facing in developing effective CAR T therapies for solid tumors, which account for 90% of all tumors. The fourth strategy is to overcome intrinsic immunosuppressive tumor microenvironment. Now we will discuss clinical considerations for early phase trials of CAR products. The clinical considerations I will be presenting here are relevant to our clinical review comments on INDs routed to our oncology branch. This slide has listed the required items of our original IND submission. I would like to note that many sponsors included a general investigational plan in their IND submission, which is very helpful for FDA reviewers. Investigators' brochure should be included if more than one study site will conduct the trial. For an original IND submission, FDA shall provide a written determination 30 days after the FDA received the IND or earlier. Now, let us review some of the key clinical considerations for early phase trial protocols. First of all, for early phase trials, the primary objective should be product safety, often using a dose escalation study to identify maximum tolerated doses and an optimal dose. During first-in-human trial stage, sponsors should make feasibility assessment of product manufacturing process. For example, the feasibility to manufacture the intended doses for intended study population, timeline for leukapheresis, lymphodepletion, and CAR T-cell infusion. Preliminary anti-tumor activity is often a secondary objective in early phase trials. CAR T therapies under investigations are mostly for patients whose disease progressed on standard therapies, similar to those approved CAR T therapies, which are indicated for refractory or relapsed diseases. During the review, we look for a number of things in the inclusion and exclusion criteria. For example, we sometimes ask the sponsor to justify some of the inclusion and exclusion criteria if they are not clear or too broad or may be inadequate. Patients with comorbidities can be enrolled if there is a reasonable justification. If the proposed study involves children, sponsors should meet requirements in the Title 21 CFR 50 Subpart D. If an inclusion criterion involves an assay of a specific antigen or HLA genotyping, the assay is likely to be considered an in vitro companion diagnostic. In this case, the FDA needs to assess whether the in vitro diagnostic poses a significant risk to the potential study subjects. We generally ask sponsors to provide an initial assessment of significant risk submitted along with their IND application. We then review the information and make a determination. In early phase trial, we now ask two key questions about the assay. First, does the assay forego or delay available and effective treatment in the standard of care setting? Second, would the assay or the results from the assay, especially false positive results, expose patients to significant risks? This slide summarizes study endpoints of different study designs. Early phase trials are generally open-label, single-arm trials intended to evaluate safety and tolerability, and to identify an optimal dose for later phase efficacy studies. Objective response rate, ORR, and duration of response, DOR, can be secondary endpoints. In the situations that single-arm trials are designed to provide primary efficacy evidence, ORR and DOR can be primary endpoints. However, I would like to emphasize that objective response means either complete response or partial response, but not stable disease, because it's very difficult to attribute stable disease to an intervention in the single-arm setting. For the similar reason, single-arm trial design is not an appropriate design for studying time to event, such as progression-free survival PFS and overall survival OS. Instead, PFS and OS should be studied in a randomized control trial setting, which is the gold standard for evaluating efficacy outcomes. Through proper randomization process, baseline characteristics between study arms are likely to be balanced, such that ideally the differences in efficacy outcomes between study arms can only be explained by intervention differences. We acknowledge that there are situations where randomized control trials may not be feasible, for instance, in some refractory patient population. However, if sponsors intend to evaluate a combination therapy, for instance, a biological product in combination with a checkpoint inhibitor, they should avoid single-arm trial design because it's very hard to discern the contribution of each component of the combination to the treatment effect. Lastly, we recognize that safety and efficacy of CAR-T therapies can be complicated and confounded by various factors, including lymphodepletion regimens, whether study subjects was treated with other standard of care treatment after the infusion of CAR-T cells, whether a bridging therapy was given to the study subject while waiting for CAR-T cell manufacturing, etc. Sponsors may consider sensitivity analysis in those situations. This slide summarizes considerations for starting dose and dose escalation scheme. Starting dose of a proposed CAR-T therapy may be based on toxicology data from preclinical studies, but we often see proposed starting dose based on doses frequently used by other trials. The latter approach may be acceptable as long as it's justifiable. In terms of dose calculation, FDA generally recommends the number of transduced CAR-T cells. This is because gene transduction efficiency can vary from lot to lot, and the best way to reduce those variations within the same dose cohort is to use the number of transduced cells. The dose can be based on per body weight, per body surface area, or flat dose. For dose escalation scheme, most common scheme is 3+3 design. However, 3+3 design may not always be the most reliable way to identify the optimal dose. More and more sponsors start to use model-based design, such as Bayesian continued reassessment method and modified toxicity probability interval. For dose escalation, we usually recommend half-log increment. FDA generally discourages intra-patient dose escalation because it complicates the safety study and makes it harder to determine the optimal dose level. For CAR-T therapies, we generally recommend at least 14 days of staggering between the first and second, the second and the third study subjects at each dose level. In addition, cohort with higher dose level should not begin until the lower dose level is considered safe. Before starting a dose escalation study, sponsors should have some ideas about anticipated toxicities and CAR-T cell expansion in vivo to help defining dose-limiting toxicities. This slide summarizes some of the DLT definitions we frequently see in CAR-T protocols. The rule of thumb is that grade 3 or higher non-hematologic toxicities and grade 4 or higher hematologic toxicities should be defined as DLTs. However, there are exceptions. We review sponsor-defined exceptions carefully and make recommendations on a case-by-case basis. For example, for allogeneic CAR-T products, acute GvHD should be also included in the DLT definitions. Sponsors should describe their CRS grading scale and management guidelines in the clinical protocol. At this point, we still recommend ASTCT's consensus grading for CRS and ICANS, published in 2019. Cytokines should be actively monitored during the first few days following the infusion of CAR T cells. Protocols often require inpatient stay as well as staying within short driving distance from the treatment center for the first 2 weeks. These are important risk mitigation strategies to prevent and manage severe toxicities after the infusion of CAR T cells. As you may be aware that for early phase trials, FDA's clinical review focuses on safety, not efficacy. We look for safety assessments that cover acute, subacute, and delayed safety events. For instance, we commonly recommend at least 28 days of DLT evaluation window for CAR products to cover some subacute and delayed toxicities. We also look for safety lab monitoring parameters and schedules in the clinical protocols. We take into account the extra safety guard achieved by modified CAR constructs. For instance, a suicidal gene expressed in the CAR, or a preemptive treatment is proposed to prevent severe CRS, or how good the preclinical toxicity data are. Actual duration of safety follow-up should also depend on the persistence of CAR T cells observed in the sponsor's preclinical studies, as well as persistence testing observed during the trial. Because CAR T products are retrovirus vector-based gene products, they meet the FDA long-term follow-up requirement. For gene products, the standard long-term follow-up is up to 15 years. In addition, long-term follow-up for replication-competent retrovirus or replication-competent antivirus may be required, depending on testing results during the first year. For more details, I encourage you to read the FDA guidance through the link shown on this slide. To minimize the number of subjects being exposed to unacceptable toxicities related to study protocol treatment, all early phase trial protocols should include study stopping or pausing rules due to safety concerns. Here are some examples. For instance, any deaths within 30 days after the infusion of CAR T cells, a death at least possibly related to the protocol treatment, increased incidence of DLTs regardless whether they are within or outside of the DLT evaluation window. To clarify, these study pausing or stopping rules are intended for temporary pause of treatment and enrollment to allow thorough safety review by the trial safety committees, DSMB or your IRB. Sponsors should share the review results with the FDA. In some cases, FDA may consider placing the study on clinical hold until a thorough safety review by the sponsor safety committee is performed and the risk mitigation plan is properly implemented. The last topic is communications with FDA. I recommend you pay attention as we have a couple of challenge questions related to this topic at the end of the talk. This chart shows you when to request a meeting with FDA. Before the IND submission, sponsors can request a pre-IND meeting to discuss product development plan, manufacturing issues, preclinical studies, proposed clinical trial design, eligibility, safety monitoring, etc. Pre-IND meeting is free of charge. We encourage that sponsors take advantage of the opportunity to gain feedback from the FDA review team. Usually, IND submissions with a pre-IND meeting with FDA are less likely to be placed on clinical hold if issues raised at the preclinical meeting are properly addressed. In the preclinical stage, in addition to pre-IND meetings, sponsors can also request two other types of informal meetings with the CBER OTAT. One is the interactive meeting, also known as pre-IND meetings, which primarily provide advices on preclinical studies that are required for initiating an IND study and product manufacturing related issues. Another pre-IND meeting is provided by CBER Advanced Technologies team, abbreviated as CAT. CAT meeting provides an interactive mechanism for prospective innovators and developers to discuss with CBER about advanced manufacturing and testing technologies pertaining to product regulated by CBER. Once the IND submission is safe to proceed, sponsors can request one end of phase 1 meeting and one end of phase 2 meeting with FDA. The purpose of end of phase 1 meeting is to review initial safety data, PK/PD data, and to identify proposed study populations for phase 2 trials. End of phase 2 meeting is a critical milestone meeting between sponsor and FDA. And the purpose of an end of phase 2 meeting is to determine the safety of proceeding to phase 3, to evaluate the sponsor's phase 3 proposal or protocol. Assuming that a product is going to be submitted for licensure, a pre-BLA meeting and other meetings along the review cycle will be held between the sponsor and FDA. I would note here that OCE provides consultation advices on all pivotal trial design and BLA applications. Here on this slide are some useful links to the informal and formal meetings. Also, this slide and the next slide provide you links to many useful FDA guidance documents for your IND applications and IND studies. The last link on this slide can take you to a page with links to many cell and gene therapy guidances. I want to give a brief summary. Although FDA has approved several CAR-T therapies, many challenges remain, as you see, especially for the treatment of solid tumors. Reducing toxicity and improving efficacy continue to be the focus of CAR product development. We, as clinical reviewers at the FDA, focus on safety reviews specific to the type of CAR product and the dosing regimens. We encourage you sponsors to communicate with FDA and communicate early. I have 2 challenge questions for you. The first question is, which types of meeting may occur prior to an IND submission? A, interactive meeting. B, pre-BLA meeting. C, pre-IND meeting. D, CAT meeting. I'll give you a couple more seconds to think about that prior to IND submission. Yes, you're correct. A and C and D. So obviously, pre-BLA meeting cannot occur before IND submission. The second challenge question is, which primary endpoint or endpoints are appropriate for first-in-human studies? First, overall survival. B, safety, feasibility, dose finding. C, landmark progression-free survival rate. D, objective tumor response rate. Primary endpoints. I'll give you a couple more seconds to think about that. Yes, correct. B is the correct answer. Safety, feasibility, and dose finding are always the primary endpoints for first-in-human studies. This concludes my talk today. I thank you all for your attention, and thanks to all of my colleagues who have given me advices, suggestions, and support. Thank you all. This is my contact information. I'll stop here.
Thank you very much, Lei and Lianne, for 2 great presentations on really very topical and interesting subjects. I think this is a completely new realm in the regulatory environment. I'm going to look through the questions here. I think we have several questions. I also wanted to welcome Dr. Peter Bross, who's also joined the panel from the same group. And I thought perhaps we could start off with a question for you, Lei, which I think is probably broad and most people will be interested in. The person asks whether you can expand on how you define early phase studies. So maybe tell us a little bit about your thoughts about early phase studies.
Sure. Thank you. And so I think for the early phase studies, it can vary significantly depending on the indication. For common conditions, such as Parkinson's disease, Alzheimer's, when we consider early phase, we're talking about phase 1 or even phase 2, which is primarily to assess safety because of the large patient population available, which allows us to do a large later phase or phase 3 trial to confirm the effectiveness and safety and for the marketing application. But in many cases, actually over 60% of the INDs we receive for the gene therapy products are usually for the rare diseases. In those scenarios, I think even for the early phase trial, as I alluded to in my presentation, we really should think the early phase trial or the first in human study as a trial which could potentially provide both safety and effectiveness data. So with that in mind, that's why I emphasized in my talk that the trial should be designed to allow assessment of clinical benefit, which we think it is important to randomize the first subject to be enrolled and to include elements such as blinding and concurrent control in the design to generate interpretable data. Thank you.
Thanks very much, Lei. I think while we're on the topic of gene therapy, there's another question which I thought might be interesting. The questioner asks whether there are any unique safety concerns associated with gene therapy trials, any special safety monitoring that has to be done in these sorts of trials, or any special follow-up. And I would add my own question, which is obviously gene therapy can be very enduring. How long do we normally look at the results of gene therapy? So perhaps tell us a little bit about the safety monitoring.
Yes, you brought up a really good question. I think for the safety issue, there are some unique safety issues related to the different kinds of gene therapy products. And for us, the gene therapy is a relatively new class of product. We are also learning as we are following these patients who have been treated either through the clinical trials or post-marketingly. So I think, for example, for the AAV type of gene therapy products, we realized that they could generate immune response post-product administration to the AAV, to the particular AAV serotypes, and also in some circumstances, the immune response also could also be generated to the transgene protein if the protein is not a known protein to the human body. And with that immune response, first of all, it would preclude those patients from receiving the second dose, and also it can trigger some other side effects after receiving the product. And for some other type of product, like the CAR-T product that Dr. Hu introduced, it has this potential insertional mutagenesis because of the viral vector's ability to insert into the human genome. So because of those unique issues, and so the safety monitoring should be designed to targeting the evaluation and potential early catch of those safety concerns. And regarding the duration of the follow-up, for those vectors such as lentiviral vector that has the capability of inserting into the human genome, the long-term follow-up guidance has recommended 15 years of follow-up. For the AAV type of gene therapy product, there are some preclinical studies suggesting it might also have the capability of inserting into the genome, although the likelihood is low. But we still recommend a reasonable duration of follow-up because as part of the questions you asked is how long the effect would last. At this point, we really don't know. So I think through a prolonged time period of follow-up, it is helpful to not only understand the safety profile, but also to understand the durability of the gene therapy product. Thank you.
Great, thanks very much for the answers there. So maybe we can turn to Peter to help us with a question here. Peter, would you be able to discuss a little bit about dosing strategies and some of the challenges for CAR T therapies?
Yeah, thanks for the question. Yeah, dosing is one of the big challenges for these types of products because, of course, they expand in vivo. So what you see is not necessarily what you get. And initially, people were really uncertain how to dose these products. We asked that sponsors plan to dose based on an analysis of the percentage of transduced cells, in other words, the cells that have been activated by the vector, and preferably transduced cells per meter squared or per kilogram. Now, we are certainly aware that some of the approved CAR T products have been approved on a flat dosing basis. So after one has more clinical experience, we can have some flexibility about the dosing. But the dosing, I think, and there are many challenges with the dosing issue, and one is that sometimes there are not really good preclinical models to base the dosing, and so mostly we suggest that sponsors provide any preclinical data that is relevant, but also to analyze and incorporate information on related types of products in terms of your first in human dose plan. And then if you intend to do multiple dosing, this should be, we generally recommend that you start with a single dose infusion and base repeat doses based on clinical experience and experience with the persistence of the cells. But it is a challenge, and I think dosing is one of the big challenges with these types of products.
Thanks very much. That's at least very fascinating to me, coming from the area of drugs. Obviously, dose ranging is a very different kind of issue. It's a lot easier to implement with drugs. While you're on the mic there, there's another question here which I think is interesting. The question is whether FDA requires randomized studies to approve CAR T therapies. I guess we realize that many of these therapies are very niche indications, and I can certainly appreciate the ability to the challenges in randomization. So perhaps you can talk a little bit about randomization.
I'm sorry, who was going to take that question?
Would you be able to take that question, Peter, on randomization?
Sure. I think that Lei described randomization early in clinical development for the gene therapy products, and I know in vaccines uses randomization early in clinical development. We in oncology with the CAR T cells tend not to use randomization early in clinical development. We usually have a number of different dose escalation strategies. The traditional is the 3x3, but we are also accepting Bayesian optimal designs and other types where you start at a lower dose and proceed and generally by half-log increments in terms of the dose increase. So we do not tend to use randomization per se in the early development of the CAR T products. And the question of whether a randomized study would be necessary for to support a BLA is something that needs to be considered on a case-by-case basis. But the CAR T cells that have been approved thus far have not been approved based on randomized studies, but there may be specific indications in which a randomized study may be required to confirm clinical benefit. But this needs to be discussed with FDA on a case-by-case basis. Did that answer your question, Dr.
Yes, it seems to. Thanks very much. So I'm going to turn to Leanne with a couple of questions here. Here is a question about guidance. Is there any FDA guidance on the clinical trial design for in-house CAR T cell production for academic centers that are trying to establish safety of CAR T cells that are manufactured in-house? So any thoughts about in-house manufacturing of CAR T cells?
Can you hear me? Actually, FDA has a CAR T cell, therapy guidance. It's coming out probably soon next year, and I don't have enough information right now. And most of the CAR T cell we received so far are autologous product, and that's what I have right now. Thank you for your question.
See, there was another sort of technical question for you, which you probably understand better than I do. The question is, are Interact meetings limited to CBER oncology products only, or can CDER oncology products also request these Interact meetings?
Yes, that's a CBER product related, and I do not know if CDER has similar meetings. And also, there's another question also asking, does CDER also has pre-IND like we have in CBER? And I encourage you to contact CBER. I'm not familiar with that. Thank you.
So this is Lei. I think the pre-IND is the same as Interact. Right now, it is a CBER program for CBER regulated products. Thank you.
Okay, it looks like we maybe have time for one or two more questions. I guess getting back to, let's have a look. Getting back to Peter, there's a question here about FDA's approach to companion diagnostics used with CAR T therapies. Do you have any insights on companion diagnostics there?
Yes, thank you for the question, and I hope that my audio is doing better because I had a microphone issue I wasn't aware of. And many of the CAR T cell products for solid tumors do require a companion diagnostic and sometimes two companion diagnostics. At least the TCR products, which are not strictly CAR T, may require HLA, particular HLA restriction in assays. And these assays have been considered for some time as part of the whole development of the product, and a positive result is required for safe and effective use of the product. And the classic example is, of course, HER2 and Herceptin for drugs, but many of the CAR T products also require a they target a particular antigen, and assay positivity may be required for study eligibility. And some of the sponsors have kind of gotten around that by saying, well, there's a high incidence of whatever it is, PSMA and prostate cancer, or NY-ESO-1 in sarcomas. And that's okay initially for dose finding, but I think these products have a definite risk, and we think that for clinical development that is optimal to develop an assay and demonstrate an association with the assay positivity and positive clinical benefit for these. And so what we ask is that the sponsors provide information on an assay in the IND, and then we review that information. And then as clinical development progresses, then we ask for more information on the clinical validation of the assay and what a positive result means, and also what the assay cut point is. And so we would ask that you discuss these with FDA at different points in clinical development.
Thanks very much. And are these assays listed in labeling, or how are they described?
Currently, assays are described in the labeling, and at least in the drug world, it says requires positive EGFR mutation by an FDA-approved test, is how the labeling goes. But this, I think, is an evolution because sometimes one particular FDA-approved assay may not be available universally. But generally, what appears in the labeling is you require XXX tested by an FDA-approved assay. That's how it's.
Thanks very much. So I think I'm going to turn back to Lei. This looks like a great question, certainly something that I would be interested in hearing the answer about. This has to do with pediatric development. And the person asks here, how much adult human data is needed before a sponsor can test a new product in a pediatric population? Can animal studies provide sufficient information to support starting in pediatrics, or do you always need adult human data first? So, Lei, any insights on pediatric development of gene products?
Oh, sure. Happy to answer this question. So in general, we recommend that the first in-human study be started or be initiated in adult patients who can understand the risk and to provide informed consent. However, we do understand for some diseases, it may not be feasible to enroll adult patients because, for example, some of the diseases are fatal and the patients may die before reaching adulthood, or sometimes the disease has been progressed to the point when the patients reach adulthood, even though they may provide some safety information, but they may not provide any information regarding the activity of the product or the potential clinical benefit. In general, we ask the sponsor to justify why it's not feasible, ethical, or relevant to initiate the first-in-human study in adults. If they could provide us with reasonable justification with supportive data, we have allowed the first-in-human study to be initiated in pediatric subjects. And of note, I think as Dr. Hu mentioned in her talk, for the pediatric subjects to be enrolled in a study, there has to be data to satisfy the requirement under 21 CFR 50 subpart D. So basically, there has to be a prospect of direct benefit to individual pediatric patients. And the data to support the prospect of direct benefit could be from preclinical studies or from clinical data, which means you may have treated adult patients first to support that, or you could use non-clinical or animal data to justify the prospect of direct benefit. And hope this answered the question.
Yes, thanks very much. I see there's a question here which, Leanne, you may be able to answer. It has to do with confounding variables on CAR-T therapy. The questioner asks, does the FDA have recommendations or best practices with regard to adjusting potentially confounding variables on CAR-T trials, e.g. prior therapies, preconditioning regimens, and or bridging regimens? For example, are there best practices for such adjustments on CAR-T protocols where investigator discretion or institutional policy applies to the bridging regimen? So I guess the question is on confounding variables in CAR-T trials. Is that something you can tell us a little bit about?
Yes. Thank you, Dr. Sachs, for the thanks for the question. Yes, I did mention in the presentation there. Some sponsors proposed bridging therapy for some patients, before the CAR-T therapy, and we generally look at the bridging therapy and also look at how far away from CAR-T therapy infusion, and also we also encourage sponsors, may need to do sensitivity analysis after that, and also if it's sensitivity analysis because CAR-T therapy may confounding the result, the efficacy result, or even safety result, and so we do ask sponsors to justify and also may do sensitivity analysis. There are a lot of confounding factors actually in these cases. Thank you.
Great, thanks very much. I think we're almost at time here, so let's first of all thank the audience very much for a great set of questions. Unfortunately, we haven't been able to get through all of them. A very sincere vote of thanks to our presenters and panel discussants, and I believe we have a 15-minute break now. We'll resume after that with the next part of the agenda. So thanks very much.